Okay, we're slowly gonna get started. I don't see any questions, but let me check just in case. Yeah, we're good. Okay, welcome everyone. This is advanced statistics, just so we're clear. I'm doing one lecture on mixed models, and Alt is doing one lecture on GLM over at basic statistics, because it helps with the more difficult subjects to do two different perspectives on it. I hear you've already heard a little bit about mixed models, is that correct? Okay, great, because that's going to help a lot. I'm just going to open the stream on my phone for a moment so I can read comments in case I'm not audible or something, and then we'll get started. Okay, yeah, so this lecture has two parts, because in the first part we already had one lecture on study design, but that was a bit limited to a certain type of study design, because all data always have to be independent. But now with mixed models we can let that go. We can include a much larger variety of studies. So first I'm going to give the second lecture on study design, and I'm going to show you all kinds of things that you can do, but that are only possible when you're using mixed models. And then in the second lecture I will explain what mixed models are, brief recap from my perspective, and then finally I'll conclude by talking about nesting, which is another thing you can do with a mixed model. Okay, so first I'll talk about balance, replication, randomization, and blocking. I already talked about replication and randomization in the first lecture, but I'll just recapitulate it because it has relevance to the second part, which is where I'll explain mixed models. Now, the important things to remember is what the difference is between a fixed and a random effect. So I will go into some depth on how you can interpret a random effect and how you can identify what is a random effect in your study. Then I'm going to talk about the choices you can make in a mixed model. So that is whether you want a random intercept, a random slope, or both, and why would one or the other. And I'll show you how to do it in R. And then finally I'll talk about nesting and how you can incorporate that using the same type of mixed model that we had before. Okay, so we already talked about this a little bit, but proper study design is very important because it increases the extent to which you can generalize the conclusions to a larger population. It increases your power, so it increases your ability to demonstrate an effect of interest. Yeah, and if you already have a pilot study, you can use that to simulate the required power, or if you know I want a power of 80%, for example, you can simulate the required sample size. So that's very useful, but it is contingent on proper study design. So if you want to generalize your conclusions to a certain population of interest, then you have to incorporate that in your study design. So if you sample people from the Netherlands, you cannot just generalize that to the entire world because we have a certain systematic difference from the rest of the world. Yeah, and if you draw a sample, you have to be aware of what it is representative of, how it was sampled, because you can't just draw conclusions about the whole human body if your sample is only on athletes, which is very common in science, where they take athletes because it's like the top performers. Let's see how this medicine works on them, or how this intervention study works on them. Same for obese people, a very common sample used in science, but of course not representative of the entire population. So you have to be aware of what you can and cannot generalize your conclusions to. So always keep in mind whether groups are comparable, because that can invalidate your entire study and make it difficult to draw any conclusions that are meaningful. Yeah, always try to randomize when it is feasible, because that prevents bias to a very large extent. And always, before you do any kind of data collection or experimenting, think about what constitutes a true replicate, and avoid pseudo-replication if you can. Okay, the first thing I want to talk about is balance. So balance means that if you have a number of groups in your study, that all those groups are equally well represented. So yeah, if possible, you should aim for equal sample size for every category that you have. And if you have multiple treatments, or you're looking at males and females and different treatments, then every combination of categories, you want it to be represented by the same sample size. It's not necessary, but this will make your analysis much easier and more powerful. Okay, if you are studying a continuous variable, it's a bit more hard to explain what balance is because there aren't groups, right? It's a range across which you can sample, but still you can have balance across a continuous variable by uniformly distributing your sample across different ranges of concentration, for example. And if you only have one observation of a low concentration and you have a lot of observations at the higher concentrations, that would be unbalanced in this case. Yeah, ideally you want your samples to be more or less equally distributed along continuous and along categorical variables. Balance is not required, and it's in many studies impossible. You know, maybe you're sampling something that simply doesn't exist as much in one thing or in one category as is in another category. Or maybe you're doing a study, you know, a clinical trial where people can drop out, people can be added at a later time. Yeah, okay, balance is not going to be possible in those cases. But if you are the one designing the study and there are no physical restrictions preventing you from having balance in your study, then you should aim for it because it will increase the power and the simplicity of your analysis. Okay, so here's what balance looks like. The iris dataset, for example, it's a little bit small, but it says Setosa, Versicolor, and Virginica 50-50-50. That's balance. That's all there is to it. Here's an example of an unbalanced design. This is from a clinical trial, and people can drop out. So here we have different age groups, and we have different amounts of smoking. And you can see that most of the groups are represented by 4 individuals, but some by 3 and some only by 2. This isn't necessarily problematic, it's just less efficient and therefore less powerful. So I just want to repeat that statement that if it's not possible, you don't have to worry that your conclusions are invalid. They're still valid, they're just not as strong. Okay, the next thing I want to talk about is replication. I see some people like my sweater. This is from Dark also. Okay, so the required sample size depends on the number of true replicates, and the things that increase the number of true replicates that you need is the variance of a process. So to what extent do individual observations differ from each other? The effect size, so if you're studying a difference that is really large, it's easier to demonstrate it, so you need a smaller sample size to do it. And the model complexity. So this one we haven't talked about yet in the previous lecture, but if you want to correct for people's sex, and you want to correct for people's age, and you want to correct for people's body fat percentage, and you have this whole list of potential confounders that you want to add to your model, those are all extra parameters. They're going to eat up your degrees of freedom, so you're going to need more true replicates to fit your model. Now, variance and effect size, you usually don't know them, but you can estimate them from a pilot study, or you can look up other studies that do something similar, see if you can find realistic values for these. And if nothing else works, you can make an educated guess of what are realistic values, and then always go towards the pessimistic side. So if you think, okay, variance could be about 2, for example, then go with 3 maybe and see what your sample size should be so that you're on the safe side. Okay, model complexity depends on what kind of comparison you want to make. So if you just want to do a t-test, your model is very simple. But if you want to do a t-test using a regression model that corrects for sex and age and smoking habits and a lot of confounders, then your model complexity is higher. So it also depends on what distribution you think the outcome has. So if you're just using a normal linear model with a normal distribution for the errors, that is very simple to estimate. But if you want to do a negative binomial GLM for over-dispersed counts, that costs more degrees of freedom to estimate because that probability distribution has more parameters. Finally, confounders. The more things you add to the model, the more you need in terms of sample size. Could I make the PowerPoint bigger? Yeah. If something falls off the screen, I'll zoom out again, but for now I can make it bigger. Oh. Sorry about that. Oh. Okay, yeah, it's okay. Yeah, I've also written about this a bit more in depth in Introduction to Biostatistics. So if you don't know any of these parameters, you cannot estimate them, you cannot get a reasonable guess, then what you should really do is a pilot study. And for that, you can determine the minimal sample size and then see, okay, is that something I can do within the time I have? Can I add a few maybe? And then you can get started at least. Okay, replication, very important subject. The subscripts fall off the screen, but you can find them in the book. The required sample size always concerns true replicates. So while we can deal with pseudo-replicates in a mixed model, we cannot increase the power of the study or the degrees of freedom that we have that way. So it does not help you meet the minimum. So again, sampling from 5 different plants is not the same as sampling 5 leaves from one plant. If I measure 10 individuals and I measure them twice, or I measure them 10 times, or I measure them 100 times, doesn't matter. I still have 10 individuals in my study. So that is very important. Determine what constitutes an experimental unit in your study, and then make sure that those there are enough of. Okay, the last thing I want to talk about before we get to the mixed model part. Let me read the question. Does similar sample size mean balance, or only totally same sample size can be better? Okay, so strictly speaking, a balanced study has identical sample size for every category. But of course, you can have an almost balanced study, and it will be almost optimally designed. So yes, balance does mean that everything is exactly the same. But if you cannot have that, then the next best thing is to have almost balanced. Okay, randomization. I'm going to show a different way to interpret this than the last lecture. But the idea is simply that if you randomly allocate your treatments, or if you randomly deal with your samples, or if you randomly sample, then you prevent bias. Now, why would it help? Let's say that you are using a spin column. This is what we used a lot at the laboratory I did my PhD research at. It's a kind of column where you can get rid of all the things except for the proteins in your sample. But the thing is, they're incredibly expensive. They cost hundreds of euros per spin column. So yeah, you don't want to use them once and then throw them away to be sure that you don't have bias, because of course we want to prevent bias, but not hundreds of euros we want to prevent bias. So we want to reuse them in a way that is minimally biased. Now, the thing is, of course, if you reuse the column, you can wash it again, but whatever you do, the efficiency of the column will slowly decrease. And a column that has been used 100 times has efficiency of, well, less than half of a new column. So that will constitute a very important difference that might confound the difference that you're interested in, of the groups you're studying, for example. So this is what that looks like. Here we have your range of experiments. You're using a column that has already been used for another study maybe. You have 20-something samples. But yeah, the first sample is going to have a different efficiency of the column than the last one, because you keep using the column, so it keeps decreasing in efficiency. This is what that looks like. And this is what it looks like if you would randomize the order in which you treat your samples. Now, randomization means that if I would just ignore the entire problem and, okay, the first 10 samples are group A, the next 10 samples are group B, then there will be a structural difference, right? You can see it. You can see that the average of group A is over here and the average of group B is over here. That is bias. That is literally what bias means. If I randomize it, then there is still a difference between the individual efficiencies that were used for the samples, but the average difference between group A and group B is now 0. And that is what we want. Okay, now we get to the interesting part, which is blocking. A block is a subset. Oh yeah, sorry.
Sorry, about the randomization. How did you then randomize? I don't understand how you go from that line to randomize. What did you do?
Okay, so that's very simple. determine the number of samples that you need for your study. In this case, we needed about 12 per group. And then you just give them a name. You name them number 1, number 2, number 3, all the way up to number 12. And the next one you can call number 13, number 14, number 16, whatever, all the way up to 24. And you just go to R, you do sample 1 colon 24, and you will get a randomly generated string of numbers that contains the numbers 1 to 24. And that is the order in which you're going to treat your samples with the spin column. Yeah? Okay, blocking means that you may have something that causes a structural difference because you have a certain grouping in your design within which you expect the experimental units to be homogeneous. Now, an example, let's say that you're doing a study on the effect of a certain fertilizer on the yield of your crops. Then if I look at a particular plot of land near a tree, that's going to be different than another plot of land which is completely in the sun all day long, and another plot of land which is near a river. They're going to be different. So if you consider those to be blocks, then that is what I mean by blocking. Here are some other examples. An experiment with several microarrays. So let's say that you need more samples than fit on one microarray. Then you should keep in mind that every microarray is not perfect, right? There are small differences in how they are shaped. They are manufactured just like anything else with small flaws in it. So it could be that one microarray is slightly thicker than another, slightly more opaque than another, etc. So you could consider them blocks. And the idea then is very simple. Same with the agricultural example. Randomize the treatments and the conditions and whatever groups there are in your study per block instead of over the entire experiment. Because that way you make sure that within every block all the groups are represented, and then the average difference will be 0 again. Now, this is one thing. This will take care of the bias, but we still have a structural difference between blocks, right? And if we could somehow estimate that difference and subtract it, then we have a more powerful study. That's what we're going to do with a mixed model. Okay, so yeah, this is the other example. If you randomly assign treatments to plots of land, that's better than randomizing it over the entire field that you have. If you have a clinical trial that involves multiple hospitals, you should randomize the treatment, so placebo or treatment, per hospital and not over the entire clinical trial. This hospital will do this, this hospital will do that. Okay, certain randomized design are better than others. For example, if you have an actual field of land, you could use a Latin square, which means that all the rows and all the columns contain all the different treatments, and it's random. I'm not going to show that here because you don't have to know that for this course. But there is a nice book by Wellham et al. Called Statistical Methods in Biology that goes a lot more in depth on these kinds of designs that are quite common in biological research, including clinical science and agricultural science and other ecological research, for example. So if this interests you, I definitely recommend having a look at the book. I can bring it here if someone wants to see it. I should have done that today, of course, but I will bring it sometime next week if someone wants to have a look at it. Okay, now why do we block, or why do we randomize within blocks? It's to reduce the bias. But if we remember which block is which, we can also reduce the variance, and that way we increase the power of our research. Now, this allows for nested and hierarchical and mixed models. Because you can estimate the difference between the blocks in a very efficient way. And that's what we're going to look at now, which is by using a random effect in a mixed model. Also, of course, if you do all of this correctly in your study, you think this applies to you, you think that there are certain blocks within which experimental units are homogeneous, then I do recommend randomizing within the blocks because it will make the analysis easier, and it will prevent any kind of confounding caused by the blocks. Now, that brings us to mixed models, which is called mixed because it has both fixed effects and random effects. Yeah, and including, so fixed effects is everything that you've learned before you learned mixed models. So is there a difference between sexes? Is there a difference caused by age? Is there a difference due to smoking behavior? And random effects is what I'm going to explain next. So including random effects allows you to model dependency. So it allows you to let go of this assumption that all observations need to be independent as long as you specify what causes the dependence in your model. Now, and the way to do that is with a random effect. And one example is the blocks in a study. You can add them as a random effect and the mixed model will efficiently take care of it. So that brings us to the important question, what constitutes a fixed effect and what constitutes a random effect? Now, a fixed effect, I'm going to zoom out a little because it doesn't fit, is a variable that has a constant effect, or we assume that it is measured without error, or it is fixed or set by the scientists, right? So the way to identify these, the rule of thumb that you can use is to think, okay, if I would measure this twice in my experiment instead of once, would the values be the same, yes or no? Well, if you within the same study, within the same time frame more or less, you don't expect someone's age to change, you don't expect someone's sex to change, you don't expect it to change what treatment they had, nor the dosage of the treatment that they had. So concentration, type of treatment, sex, and age, those are fixed, right? In practice, they are of course not measured without error. Nothing is measured without error. Because you may think that you applied 100 milligrams, but in practice it was half a milligram less, or it was half a milligram more. But we assume it to be 0. That's the important part. Same thing, if you measure 1 year later, yeah, of course you're not the same age, but within a certain time and location, you would expect these variables to be constant. Okay, what is then a random effect? A random effect is something that introduces dependence in your data. It introduces some sort of structural difference. And an example is patients, because you measure the same patient twice, like before and after some treatment. Then the measurements from the same patient, if the patient started out with a low blood pressure, they're going to end probably with a lower blood pressure. Yeah, and that way it introduces kind of correlation in your data that you don't want, which is called dependence. If you include students from multiple schools, then schools is a random effect, because one school may have a really rich person funding it and has millions of euros, can buy whatever material it wants. Other school might have to do with limited resources, like Leiden University. But that means that there is a structural difference between the students you sample from one group and from another group. I'm, of course, kidding. Leiden University is the best. Another example, if you're doing a meta-analysis, meta means about itself, so a study about studies, that is the ultimate way to get an idea of what does science think at the moment about X. So, for example, is too much salt bad for you? This is a typical question that has been studied many times. So why not combine all those studies? Why not combine all the results? And you can do that, but within a study, the same scientists were present, the same equipment was used, the same samples were assessed. So that introduces a structural difference that you should account for with a random effect. Now, observation from the same patients, schools, or studies, we assume them to be more closely related to each other than to any other particular observation. And if measured twice, you would want them to be different, right? If you have the potential to sample from a larger group of patients, you would use different patients. You wouldn't use the same ones again and again. And if you want to know, okay, is this book better than that book, and you're going to give students one book or the other book, you're going to let them do an exam, and you're going to look at their grades, then of course you want to include as many schools as possible, because you want to get rid of the effect of schools, because that's not the question you're asking in your study. The question is which book is better. And of course, if you have a meta-analysis, you don't want the results to be completely contingent on one really important study. No, you want to include as many studies as possible. So if measured again, you would want to have different observations. Now, the effect of these variables, we assume them to be on average 0. So on average, all the schools have a difference of 0 from your model, but individually they differ. Now, we could estimate a fixed effect for every different school. Let's say that you're assessing 100 schools in the Netherlands, but that costs 100 degrees of freedom, which is very inefficient. And also, what if you want to generalize your conclusions to school number 101? Your model cannot do that. But if you estimate a random effect, then instead you estimate the variance between schools. And then you can generalize the conclusions to any other school, because you just say, well, we expect it to be here, but because schools differ from each other, there's not just the residual, but there's also a slightly larger deviation from the variance between schools. Yeah, so that's what we're going to do. We're going to estimate the variance that's caused by the random effect. Now, there are exceptions to the rule. The rule of thumb is nice, but it's just a rule of thumb. But if you're interested in a specific set of patients, of course you're not going to measure different patients, right? So if you want to write a report on what went wrong in a certain clinical trial, you can't have different patients because that happened in one clinical trial, right? So in that case, it is a fixed effect and not random. Same thing, if you want to compare schools, then the particular schools you're comparing are the ones of interest. So you want a fixed effect, way easier to interpret. You can do comparisons and do a t-test and stuff within your linear model. So in that case, you want a fixed effect for which school it was. Same thing, if you're not doing a meta-analysis, but you're just contrasting two specific studies, you're saying, okay, this study said this, but that study said this. In that case, it's a fixed effect because you're talking about two specific studies and not some larger possible population of studies that could have been performed. Now, in this case, it would be fixed. In all the other examples I gave before, it would be random. Okay, simple example of what the model looks like is like this. This is a simple linear regression, but I've added something, right? We have beta 0, which is the intercept. We have beta 1 times x, which is the slope times the explanatory variable. I've added something. I've added epsilon times z. Why is it called epsilon and z? I don't know. Other characters were already taken. Just remember it. And epsilon is the residual we already had. But this is what it looks like. Not super important, but it is useful once we see the output, because then you can see how you can add this to your predictions. Okay, I'm quickly going to read the questions. Why do you group by schools in the first place? Can't you just pile them all into one big group? Okay, yeah, that brings us back to the problem of blocking, because within one school, we might expect students to perform better or worse. You know, if the school has better teachers, the students are going to do better. So there is a difference between schools. So of course you can just ignore the schools and pool them all together, but then your study is less powerful because you didn't correct for the fact that different schools will perform differently. So it's like confounding. And make it fixed, losing a few degrees of freedom. Yeah, okay, a few degrees of freedom. That depends how many schools. If I want to assess all the schools in the Netherlands, then it's going to cost a lot of degrees of freedom. It means you need a really, really large study to efficiently model it. And the other thing is, don't spend more degrees of freedom than you have to for inference, because if you have more degrees of freedom left, the estimates will be better. They will have smaller confidence intervals. They will more likely be correct. So yeah, you don't want to use degrees of freedom if you don't have to. Okay, yeah, we can also do this. This was a mixed model with a fixed and random part that is simple linear regression. With multiple linear regression, it looks the same. You know, that's just multiple linear regression. Intercept plus slope 1 times explanatory 1 plus slope 2 times explanatory 2, etc. Just add it to the model. Looks the same, except there's the random part over there. Now, what is this random part? It is actually quite similar to the error term, yeah, except that we multiply it by z, which is the random effect. So in this case, could be school. So that's a matrix of zeros and ones indicating, oh yeah, it was that school. Oh no, it was that school, etc. So it tells you where to add the effect. Yeah, how do we do that? We just estimate another variance, namely that of the random effect. Okay, this is the mathematical way to describe it. I imagine not everyone is now able to follow what that would look like. So next we're going to look at what this would visually look like. But first I'm going to read some questions. It's a bit fast for me. Okay, sorry, I will slow down. It's a problem. I understand. Is there a standard way for calculating degrees of freedom in fixed and random effects on the model? Good question. There is a way to do it called Satterthwaite's approximation. It is not easy. So for a fixed effect model, it's very simple, right? We just count all the betas. Yeah? So in this model, if it were a fixed model, so we didn't have this part, this would have 2 degrees of freedom for the model. This one would have p degrees of freedom for the model because we have p fixed effects. Oh yeah, sorry, and an intercept, so we have p plus 1. The random part is not just one thing we're estimating, but it's a variance, and just like with the Welch t-test, it is really hard to express how many degrees of freedom it costs to do that. It is less than the fixed part, so it's not much, but the exact calculation we're not going to cover in this course. So the short answer is yes, there's a standard way, but I'm not going to show it. Okay. There's subscripts over here, so it says sigma squared epsilon, sigma squared epsilon. The only reason for that is to show that these are different variances, so not the same thing. But enough of the notation. Let's see what it looks like. Here we have an example of 12 patients in a clinical trial on chronic hypertension. I'll make it a little bit bigger. And observations of the same patients are, of course, correlated, and you can very clearly see that because the legend, I'm very sorry to colorblind people, there doesn't exist a color scheme of 12 colors, all of them uniquely identifiable and colorblind friendly, but you have to take my word for it. This shows 12 different patients, and you can see that they have been measured over time. So this brown observation has been measured several times. It's the highest one, and you can see this person started out very high and ended up very high. So these measurements are somehow correlated to each other. Yeah, and you can see the lowest one also started and ended as the lowest one. So you can very clearly see that this, if you don't account for this, then it's going to invalidate your conclusions. So let's fit a mixed model to that and see what happens. This is what a linear mixed effects model looks like. I will go over this completely, but I cannot make it much smaller, so I'll just read out loud what I'm looking at. So every patient has a slightly different value overall, and by estimating this random variance, we're going to correct for that. That's the idea. Well, how can you do that? Okay, first, this is not quite linear, because it doesn't make sense that blood pressure would forever keep going down as the weeks progress. They would all die. So let's do logarithmic instead. That's a bit safer for the patients. Their blood pressure will plateau, right? That's what we want. So we have the blood pressure as an effect of the logarithm of time, yeah? And I added 1 because you cannot take the logarithm of 0. So I just do plus 1, but that's just correction, doesn't matter. Then the random part in the package that we're going to be using in this course is specified like this. This means I want a random intercept. Not super important now because I will explain later what random slope means in the choice. For now, this is what a random effect looks like. It's 1 vertical bar patient. That means estimate a random offset for the patients for me. And that's what the model does. So first it says what it did. So it said, okay, this is your formula. That's what I've written here. This is the data that I'm going to get these variables from. And then it says Remmel criterion at conversion. So it starts with the worst thing to explain. Remmel is restricted maximum likelihood. That is the way that a linear mixed effects model is estimated. It can also be done with something called the ANOVA method, which is another annoying name because it has nothing to do with ANOVA. But this criterion, usually you can ignore it. So it doesn't matter much what it says, unless you get an error or a warning. If your model did not converge, these are more complex models, so you will run more often into convergence errors and problems and warnings. They're more difficult to work with. And then you should look at the criterion. The exact number doesn't matter, but what you're going to do, something went wrong, so you're going to simplify your model, or you're going to scale your variables, or you're going to transform your variables in some way, and then the criterion will change. It might still not converge, but if it's changed in the right direction, so if it lowers the criterion, then apparently what you did was correct. So then you're going towards the right model. So if you simplify your model, criterion goes down, then okay, your model was too complex for the amount of data you have. That's what it's for, but yeah, we're not going to use it in this course. I just want to go over the complete output of this model. Then we have the scaled residuals. Now we've seen this before in the fixed effects model, so all the other models we've had so far. This just shows, you know, where your residuals are. The lowest residual is minus 1.95. The highest residual is 2.8, more or less. Yeah, the minimum and maximum. Then we have the first quartile, the medium, and the third quartile. So that's the first 25% of your data, first 50% of your data, and the first 75% of your data. Why does it show this? For people who are too lazy to do diagnostics, because you can kind of see if normality of the error term is approximately correct. Because if your residuals follow an exact normal distribution, then it will be symmetric, and then the mean will be equal to the median, which is 0. So if the median is very far from 0, then apparently normality is not a reasonable assumption. But yeah, now that I've explained what we're looking at, you can forget it again, because we're going to do diagnostics, right? Which is much more informative than just these 5 numbers. So it's just there for, yeah, for reasons I don't really understand. Then we have the random effects. I'll explain that second, but first I'll explain the fixed effects, because this you're already familiar with. This is no different than the linear models we've been looking at so far. We have an estimate for the intercept of 150. So that means that this model says that, okay, we're going to estimate a logarithmic line that starts at 150. And for every increase in the logarithm of time, we're going to subtract 10. Yeah, that makes sense, of course, because it's supposed to go down over time. Yeah? That one you already know. Standard errors is a measurement of uncertainty of these estimates. T-value, you can use that for a test, see if the effect of time is significant or not. Now the different part for a mixed model, the random effect. The random effect is this. This is the important number, this 17.5 something. So we have the residual variance. You also have that in any other model, right? That just means the extent to which observations differ from the linear model you estimated. But this one has something else. It has a variance for patients. Now, you can very easily see what is going on in this model if we look at this again. Over here, we have the residual variance. It's very small, right? It's 0.8. And we have the patient variance, which is 17. It's much larger. Okay, how can you interpret that? That means that if we estimate a logarithmic relationship for the effect of time, then there is a small deviation that patients have to their individual logarithmic line, but the difference between patients is much larger. You can kind of see that that's true, because if you imagine a logarithmic relationship running through this brown set of observations, then you know the individual observations are quite close to each other, right? But if we look at the different patients, that difference is much larger, yeah? And of course, variance is a bit difficult to interpret, how large it is. Much easier is the standard deviation, because for things we assume to be normally distributed, we expect things to be more or less within 2 times the standard deviation. So now we can actually check that. We would expect everything to be within about 8 from the mean, right? Because this says 4, and 2 times 4 is 8. So that means that the range of patients, we expect 95% of all patients that we would conduct this study on to be within 8 of the actual intercept. And that intuitively, that looks more or less correct. They are about within 8 of 150. Now, the residual standard deviation is about 1. That's much smaller. So that means, okay, all these patients differ from each other, but within a given patient, we expect a maximum difference of about 2 from the logarithmic relationship. Yeah, that's what it shows. Okay, that's almost everything in the output. The last thing is the correlation of the fixed effect. That one we're not going to use in this course, and is not super important if you already, carefully chose your model. So we're going to ignore that one. If I'm still going too fast, please comment again, and I will slow down further. Okay. We can do a little bit more with the random effect to visualize what's going on. So the way to do that with this package we're going to use is to call the function ranf for random effect. If I do that, it will show me the individual deviations that these patients have from an average distance of 0 from the intercept. And okay, it says -3, 3, -1. Okay, let's use the colors again. We can see that, okay, apparently this brown patient has estimated to have an offset which is the highest of all. It's about 9 or so. And this green patient is the lowest of all with an offset of minus, what's that, 7. Let's see if we can find that. Okay, apparently that's patient number 4, that's the green one. And patient number 12, that is the brown one. Now, what are these distances? That's what we see over here. But the cool thing about a mixed model is one thing that people who perhaps don't know about mixed models do is they just calculate what is the value at baseline and they subtract it from their measurements. Very inefficient. I don't recommend it. Because this mixed model, it doesn't only look at this. It looks at the entire range. And then it estimates, okay, across the whole range, how much does the brown patient differ from the blue patient, differ from the lilac one, differ from the yellow one, etc. And that is much more efficient. Okay, so that's what the random effect looks like. Then we can plot them next. This is much easier. We can put them next to each other, and you can see that indeed the highest one is the brown one, is over here, and the lowest one is the green one, is over here. What did this model do? It estimated a normal distribution with a mean of 0 and a variance for the patients. And remember what I said earlier, it's not just that we're using less degrees of freedom, but now if I have a patient that has never been measured, I can estimate where they would fall on the line by saying, okay, the random effect is, the average is 0, so I will say 0, and I can express the variance of this random effect to predict how far from the relationship this person will be with a certain percentage of confidence. Okay, and we only used one effect. We only estimated one variance instead of estimating 12 different offsets for all the patients. So this is much more efficient than a fixed effects model. Why is there no p-value? Yeah, we'll get to that. Hold on. It takes into account the patient differences, and I have to zoom out again. Sorry. That means that inference from this model is valid even though these are not independent measures, and that is why we're using a mixed model. Okay, what does a random effect look like? This is what it would look like if you plotted your model. So we have a logarithmic relationship for time, and we have a random variance, so that means that every patient has an offset from the original intercept. So every person has their own intercept and then progresses over time. Yeah, so this is what the model looks like. And again, we can see that indeed individual observations don't differ much from their relationship, but the difference between patients, difference between colored lines, is much larger. That's also what we saw in the output. Oh, oopsie. Okay. Then we get to the choices you can make in a mixed model. So the most important choice is whether you want a random intercept or a random slope. So a random intercept is like saying that there is some baseline offset for the category, but everyone progresses the same way, right? A random slope is saying, okay, everybody starts out the same way, but there is a different progression over time. So this is what that looks like. A random intercept is, okay, for every patient, for example, we would have a different starting point. A random slope is saying, okay, there is a certain effect of the explanatory variable in the outcome, but there is a variance to that effect. So every person will progress slightly different. If you can do one, you can do the other. You can, of course, also do both, but in practice, you will very rarely use both because it eats up a lot of degrees of freedom, and you'll most likely get convergence error. Okay. Yeah.
The choice between these three, does it depend on the biological question you're trying to
Yes.
answer, or would you just try them and then test which model?
Okay, that's a very good question. I don't know if it was audible, so let me repeat it. Does the choice of random intercept or random slope or both, does it depend on the problem you're studying? Of course. You should try and think about which of these makes more sense from a biological point of view. However, a random intercept is much easier to estimate than a random slope. So in practice, I usually start with a random intercept, and only if I have a lot of data would I even consider using a random slope, because it's just more expensive in terms of degrees of freedom. And then you might wonder, okay, is it then still correct? Of course, you would preferably have the model that makes the most sense, which in many cases is actually this one, of course. But you know, if you can't, you go with the next best thing. And the cool thing is that doesn't matter which of these three you go for, all of them take care of the dependence in your data. So all of them will turn a comparison that you cannot do with a normal linear model valid with a mixed effects model. Yeah? Why is the random slope more expensive with degrees? Yeah, sorry, that's a difficult question that I cannot answer concisely. It has to do with the way they are estimated, but that's unfortunately not part of this lecture. But if you really want to know, I can show you after the lecture. So the question was why does this cost more degrees of freedom than this? It has to do with the variance covariance matrix and what exactly you are allowing the model to estimate. And it turns out that the second scenario it allows for more different things to vary than the first scenario, but it's a bit beyond this introductory lecture. Okay, what does a random slope like in our previous example? We already saw what a random intercept looks like. That's like this. We allow them to vary a fixed amount from each other. Then what does a random slope look like? Anyone have an idea what it would look like?
They start at the same point and
Yeah.
they increment at different rates.
So where the explanatory variable is 0, they will start out the same, but they will have a different progression over time. Yeah, so this is a random slope. Okay, patients start out equal but develop differently over time. In this case, doesn't look very realistic. The data suggests otherwise, right? Because they look quite different over here, and you know, these lines, those curves, sorry, that are being estimated, don't really look like what we see in the dots. So it's not a good model, in my opinion. So what does having both look like? Anyone have an idea?
They all start out different places and they all slow differently.
Yes, they all start out differently and they all progress differently. So let's see what it looks like. It looks more or less the same than the random intercept, right? Looks more or less like this one. So why is that? Anyone have an idea? Why does it look more or less the same than just random intercept? Yeah. So you said there was only a standard deviation of 1 in the residual. That's right. That's what it has to do with. Let me show you precisely why. This is the output of that last model I showed. And we're just going to look at the random effects part over here. We have the intercept, the random intercept. So this is the variance in patients as a constant difference between them. And we have the residual variance. So the extent to which people differ from the model is much slower. Is much less. Only 0.8 is left. Then we have the variance in the way they progress, and that is yet another 400-fold less than the variance between individual observations, which in turn is about 10, 20 something fold less than the variance between the patients. So why do they look the same? Because the different ways they progress is very, very, very subtle. It's very small. So in cases like this, of course you want a model that most accurately represents the biological situation. But any model is a simplification of the actual process. Any model is wrong. We just want a model that's useful. And in this case, if we remove the random slope, it looks almost the same. So we can have many more degrees of freedom left, have a much better estimation, and use much fewer degrees of freedom by just removing this random slope. So that's why I want to show you, in practice you very rarely use these types of models because they cost too many degrees of freedom. So it's a practical consideration. Okay, sorry, let me see what I wrote down there. Yeah, is it really necessary? The answer is no. Yes?
Do you start with the most complex model here or the simplest model?
Okay, questions. Do we start with the most complex or most simple model? I would start with only a random intercept or only a random slope, depending on which one makes more sense for your study. I would not start with the most complex model because most cases it just doesn't converge. And then, yeah, how do you continue? I would also not start with the simplest one because usually both the intercept and the slope will run, and then you should go with the one that makes the most sense and not the one that has the best AIC value or something. Okay, yeah, a mixed model with a random intercept is very efficient. We don't really go into the details of the math of why it is efficient, so just take my word for it. But if we only have to estimate a variance in the slopes, in the intercept, it is very cheap to estimate, very simple to do, so it's a very efficient model, has a lot of power. If you use a fixed effect, you would have to estimate a separate effect for every category, so it costs way more degrees of freedom. Now, random slopes allow for very interesting models. You know, differential progression over time is something that could definitely make sense in your study, but be aware that this is a more expensive thing to do, so you need more true replicates. Yeah? And if your model is more flexible, that by definition means it is more prone to overfitting. So even though yeah, it might seem appealing, it is not always, practically speaking, an option. Okay, how do you do it in R? I already showed it a little bit with the previous example, but let me just show you the notation. There's a package called LME4, which is really one of the best mixed models packages in R, and you can use it like this. Linear mixed effects regression, LMER, and then the fixed part looks exactly the same as you would for a normal linear model. So you can add things like x1 plus x2, you can have interactions like x1 times x2, works the same. Then the random effect is specified like this, between brackets 1 over the random effect. So this would be patience in the example I just talked about. Now, random slope, you have to say 0, which means no intercept, plus the thing you want a random slope for. So if progression over time is the thing I want to differ, then that is what I estimate a random slope for. So 0 plus x, vertical bar, z. That will give me a random slope. If I want to do both, I can just say x over z, because the default is to include an intercept for reasons we're not going to go into now. And that means that it's the same as saying 1 plus x, because 1 then means intercept, and x is the thing we want a random slope for. Yeah, you've also had GLMs in this course, in the third year course. GLM, you specify the family that your, the distribution that your outcome belongs to. So is it a count? Is it a ratio? Is it binary? Is it normally distributed? You can just add that as an argument to the function generalized linear mixed effects regression. So just add a G and add a family, and then you have a GLMM. I make it sound simple, but in practice these models are really hard to work with, and they will not work for reasons that will elude you. But within the context of the course, this is all you need to know, and if it doesn't work for some reason, we're not going to troubleshoot it because yeah, this is just a short course and we have a couple of days to do this, so we're just going to show the cases where it does work. If you need it for your internship and it doesn't work, just send us an email. Okay, so they are harder to work with. So yeah, I definitely recommend consulting us or someone else if you're working with these, but implementing them is very simple, right? It's just the things you already learned with GLM and then add the random part. That's all there is to it. And that's also why I like this package, because it uses that same language that base R already uses. Okay, so a mixed model can also account for much more complex structures, much more complex ways that make your data dependent. And this is the last part of this lecture, which is nesting. So we already talked about clinical trials. Let's say, you know, there is a pandemic and we want to do a clinical trial in more than one country. Then we have different hospitals from different countries, and the patients are nested in a hospital, which may have better or worse healthcare, which is nested in a country, which may be richer or poorer or may have different rules. So that means that there's a nesting structure, a hierarchy by which your data are dependent. Now, that's what we're going to cover now, how you can do that with a mixed effects model. Another example from agriculture, let's say that you want to compare two different ways to grow your crops, two different treatments or so. Then if you include multiple farms in your study, of course that's better, because then you can generalize to any farm, right? But if a farm has multiple plots of land, one near a river, one near a tree, etc., then they will differ. So you have the plants that grow within a certain plot of land, which grows within a certain farm, and the farmer has his own way of doing things. So there is a hierarchy to which your data are dependent. Here's a visualized example. Let's say that we want to test a standardized exam across the European Union. So we already have CITO in the Netherlands. Let's say we want a European CITO, because we all love the European Union very much. What we could do is include multiple countries. Probably a good idea, right? Because we generalize it to all of Europe and not just to the Netherlands. But different schools, may have different material available, and different classes within schools have different teachers. Maybe one teacher is much better than another, maybe one is more enthusiastic than another, and then it will cause some difference in the end in the extent to which the students were able to learn or exposed to the material they have to learn. So all those things affect the outcome in a hierarchy, because teachers are, a class is in a certain school and a school is in a certain country. That is what nesting is. Okay, then how do you model that? Fortunately, that part is not very difficult, because the mathematics of this is really difficult. We're not going to show it, but in R, it will automatically take into account on which level in the hierarchy something takes place. So you have a certain treatment and you're like, yeah, but I didn't apply treatments to students. Not student A gets test A, student B gets test B. No, I did it per school. Is this study still valid? Yes, it is, because R will automatically see where the dependence is if you specify this correctly. So it's very simple to do. It's just a bit more hard how it actually works. So the way to do it is to start by specifying the uppermost class of random effect. So in this study, what would that be? Countries, exactly. Because that's the highest level of this comparison. Now, then, after specifying the superclass, so the highest class, you specify the subclass, so the one that is nested within it with a slash. This is if you're using this package. Other packages may work differently, but in this course we're only using this one. So this would be country slash school slash class, right? Because that's the order of nesting. And that's what we see here. We have grades, for different type of exam that we're studying. I said textbook, but it could also be an exam. And then we have a random intercept, which is what I would start with if I didn't know anything else about this study, because it's the cheapest one to estimate. And then we have country, and nested within countries we have schools, and nested within schools we have classes. Now, then the question I often get is, why does it not say slash student? Does anyone know the answer? Yes. Yes, exactly. We're measuring within a class, not within a student. In this one, I have written down patient, right? We have some clinical outcome. I ran out of originality. We have a treatment effect, and then we have the country within it took place, and the hospital in the country, and the patient in the hospital. That's strange. I just told you I didn't add student, so why did I add patient over here? Let's say it's the example I showed before with the 12 patients progressing over time. Yes?
Because the treatment happens within the patient.
Exactly, because we have more than one measurement of patients. So we have dependency within patients. If I only give one type of exam to one student, then I've only measured the student once. So student is the residual, right? So it's not part of this structure. But if I measure patients before and after a treatment, then I have measurements within patients, multiple times, which are within hospitals, which are within countries. Yeah, so that's how you specify this random nesting structure. Okay, I have some questions. Does nesting model always relate to certain places? No. You can have nesting with other things than physical hierarchy, right? Let me think of a meaningful example. Let's say that I come back to that later because I can't think of a good example. But it's just easiest to visualize it if it's in a physical place. It doesn't have to be. It could be that it's some other thing that is causing the dependence. I do have an example. Let's say with the patients, we measure them multiple times. That's not a physical place. It's just the same patient. The patient can move around. I don't care. But they are repeated measures. They are nested in the patient, the measurements. Yeah, so it can also be temporal, and I will try and look up some other examples to come back at this question later. Okay, because there are no true replicates for students. Yeah, I'm sorry, I was only looking at the class, for instance, but that is correct. There are no true replicates for students in this study because we only measured every student once. In the patient example, if you measure them multiple times, they are a random effect. Okay. This is a self-test. We can discuss this on Monday. So I will also post this on Brightspace. I think I already posted the Word version, but I'll check, of this lecture. These are some examples, and you have to identify whether what the fixed and random effects in this study could be. Of course, these are very short sentences, so I didn't explain the complete study. Just be creative, just think of, okay, maybe they were measuring this and it was in this way. Describe it and then tell me what is a fixed effect, what is a random effect. We can talk about that on Monday or some other time. I'll have to check the schedule when I don't have to be at basic statistics, and otherwise I can, you can ask questions about this on Brightspace and we can discuss it there. Okay, then short summary. We talked about some consideration of experimental design that tie in with the use of mixed models. So particularly blocking, where you can do 1 over block, and then you have corrected for the fact that there are different blocks. The point of this is that if you design your study well, even if you're not really good with mixed models, trust me, in the end it will save you time. It will save you time and it will give you a better conclusion. So prevention is superior to cure. The real takeaway from this lecture is the part about study design. And the mixed effects model, of course you need to know it for the exam now, but if you forget about that 1 year from now, that's okay. You can ask for help. But the study design, that part you should get right or ask for help before you collect data. Okay, so if there is any kind of dependency, block dependency or repeated measures or any kind of dependency in your data, you need a mixed model. Remember that pseudo-replication can also be used, by having a random effect for the last experimental unit that was measured multiple times, but it doesn't increase the number of observations you have. So be aware of what constitutes a true replicate in your experiment. And then a mixed model can handle data both with true replicates and pseudo-replicates more efficiently than if you would just take the average of your pseudo-replicates. Yeah, so it's a very powerful model, the mixed model. And we see it a lot in biology and in clinical science. So it is very useful if you're just aware of what it is and when you need it. That after this course, that's the most important. And during this course, it's up to the boss what you should remember. If you want to rewatch part of this lecture, you probably know that most of the lectures are on YouTube, but I just want to show that I have chapters over here. So if you just want to remember the nesting part or something, you can go over here and you can see where I talked about what. Okay, are there any questions for now? Yes. I measured a lot of what?
In the nesting model,
Yeah?
doesn't you use a lot of digital freedom?
So your question is whether this costs more degrees of freedom? Yeah. So of course it costs more degrees of freedom than leaving it out. But remember, the reason we're doing this is not so much for efficiency purposes, but to model dependency. Because I could just omit this, but yeah, then I would have a model that gives incorrect standard errors and incorrect p-values and incorrect confidence intervals because there's dependency in the data and that violates a fixed effect model's assumptions. So it's a necessity. We need this. And modeling it in this way, yeah, that allows us to take care of a problem. Yeah, and the second question is about efficiency, and then usually it still turns out it makes models more efficient, but that's not always the case. Yeah? Yes?
With this example of the 20% school in the class, so with the nesting model at the bottom, you would not add class as a subclass. You accelerate. That's what I'm questioning.
Sorry, I did not quite follow.
The random effect superclass was country, and the
Yeah?
random effect subclass was then school, but you didn't add class, right? Is that what I understood correctly?
Depends. If I only take one student out of each class, then I would not include that as a random effect. But I'm sampling multiple students from each class. So students nested in class, nested in schools, nested in countries. Right? So that is the nesting structure in this case.
So then you would add class, not student.
Yeah, exactly. The last thing in the chain of nesting is the thing you don't include because you only measured it once.
But what if you measured the one patient multiple times?
Then you add it.
But then it's the last thing, right?
Yeah. Well, no. The last thing is the individual measurement. Yeah. So that's why I used physical example because they're easy to interpret. But from a statistical point of view, the last thing you measured is the measurement itself. Yes.
How would the output of this look?
I'll leave that to Harald. No, sorry, I don't have simulated data here with nesting in it. Yeah, it doesn't look much different. I can't tell you what it looks like without showing. So here we see patient, right? It has an intercept. If patients are nested in hospitals, then you would also see a variance for hospital. And you would see like hospital : patient or something to indicate that it is inside the other one.
So you get an estimate of the variance.
Yes, exactly. You get multiple estimates. Yeah. Okay, if there are no more questions, or if there are more questions, actually I should say, please ask them to Harald or post it on Brightspace because I have to run to the other building. Yeah, I hope it was somewhat clear, and if not, you can ask questions later.
Okay, well, can you keep it on for a moment?
Yeah.
I think it's almost 11:30, and I have to check out how it will work out with the recordings. So I would suggest that we would start with the first assignment of today, which is actually analyzing a nested mixed model. So we do that first. What we could do after that is that we start a little bit earlier with the afternoon lecture. So let's say that we could start with the afternoon lecture. What was normal? 1:30? 1:30? So 12:30? Yes? Oh, that's a yes? Okay, I see a yes. Another yes. Okay, 12:30. Then I will talk about simulation, and actually I will simulate a nested model, so you could see the outcome. And then I will give an overview of this whole week and the week before, so you can see the different structures on which we build. We start with a simple model, then it becomes more complicated. So I want to put that in one lecture using the blackboard. Yeah? Okay?
Okay, I'm gonna race to the other building.